Fine, let's start. Number 1. The Guinea Worm. Imagine a blister rising on your ankle, and then imagine that blister splitting open to reveal the blunt head of a living worm. A pale thread that keeps coming and coming until nearly a meter of it hangs from your skin. This is Dracunculus medinensis, the guinea worm, and it has been erupting from human bodies for thousands of years. The adult female grows between 60 and 100 centimeters long, yet she is only 1 or 2 millimeters wide, roughly the width of a strand of spaghetti. The name of the disease she causes, Dracunculiasis, translates to the affliction with little dragons. The species name, medinensis, points to the old city of Medina, where the worm was once a familiar horror. You do not catch this parasite from another person. You drink it. In stagnant ponds, tiny water fleas, called copepods, carry the worm's larvae. When a thirsty person swallows that water, stomach acid dissolves the copepod and sets the larvae free inside the body. There the worms mate. The male dies quickly and quietly. The female begins a slow migration through the connective tissue of the body that lasts about a year, until she reaches the skin, almost always in the lower leg or foot, and burns her way out. The ancient treatment is the strangest part. For at least 3,000 years, healers dealt with the emerging worm by winding it, a few centimeters at a time, around a small stick, over days or even weeks. Pull too hard and the worm snaps, and the wound turns septic. That patient, careful winding, is still, essentially, the modern method. There is no drug that kills the worm, and no vaccine that prevents it. A calcified guinea worm has been found inside an Egyptian mummy dated to roughly 1000 BCE, one of the oldest physical parasite specimens ever recovered from a human being. Here's where the story reaches into myth. Some scholars argue that the serpent coiled around the staff of Asclepius, still the symbol of medicine today, began as an image of a worm wound around a stick. Others connect the fiery serpents that tormented the Israelites in the Book of Numbers to guinea worm outbreaks near the Red Sea. The Greek physician Galen wrote about the condition, but mislabeled it a disorder of the nerves, having never seen a living case. For centuries, physicians called it the Medina vein, believing the worm was a diseased blood vessel. Because the parasite has no important animal reservoir, it can be wiped out entirely. A campaign led by the Carter Center cut cases from an estimated 3.5 million a year in 1986 to fewer than 15 human cases a year by the middle of the 2020s. If it falls, it will be the first human parasite ever eradicated, and it raises the question this whole video circles. What if the true rulers of prehistory were never the kings, but the things living inside them? Number 2. Chagas disease in the Chinchorro mummies. On the desert coast of what is now northern Chile, a people called the Chinchorro were preserving their dead through artificial mummification around 7,000 years ago, more than 2,000 years before the Egyptians began wrapping their own. And inside these mummies, scientists found something no one expected: the oldest molecularly confirmed parasitic infection in all of human history. The culprit is Trypanosoma cruzi, a single-celled protozoan with a whipping flagellum, the cause of Chagas disease. It is carried by blood-sucking insects known as kissing bugs, which bite sleeping people around the face. The disease works slowly and cruelly. The parasite invades the muscle of the heart and the digestive tract, and decades after the first infection, it can produce a swollen, failing heart, a grossly enlarged colon, and an enlarged esophagus. In 2004, a landmark study led by the paleopathologist Arthur Aufderheide tested tissue from roughly 283 mummies from northern Chile and southern Peru. He found Trypanosoma cruzi DNA in about 41% of them. The oldest infected individuals date back some 9,000 years. Across that vast span, from ancient foragers to modern populations in the same region, The burden barely changed. Roughly 4 in 10 carried the parasite the entire time. The detection was a triumph of a young science. Researchers used PCR to amplify a specific sequence of the parasite's kinetoplast DNA, proving its presence in tissue that had lain in the desert for millennia. That the DNA survived at all is thanks to the Atacama, the driest desert on Earth, which preserved soft tissue for 90 centuries. Kissing bugs like to colonize the thatch and mud walls of houses, so when humans began living in fixed dwellings, they accidentally built the perfect home for the insect that spread the disease. The parasite is found only in the Americas because its insect vectors are New World species, and it circulates naturally among opossums, armadillos, and rodents. That means Trypanosoma cruzi was here long before people were, co-evolving with American mammals for what may be hundreds of thousands of years. Even Charles Darwin is suspected of catching Chagas from a kissing bug in Argentina during the voyage of the Beagle, an infection some believe explains his decades of mysterious illness. The disease is named for the Brazilian physician Carlos Chagas, who in 1909 described the pathogen, its insect vector, and the human disease all at once, a feat almost unheard of in medicine. Modern Chagas still infects 6 to 7 million people and kills tens of thousands every year. What the Chinchorro mummies proved is that the so-called diseases of civilization did not wait for cities. This one was already hollowing out hearts among people who lived by the sea and buried their dead in the sand. So if disease reached even the wandering coastal foragers, what did it do to the way our bodies were built? Number 3. Clothing lice. Humans carry 3 kinds of louse, and one of them can answer a question that leaves almost no trace in the ground. When did we start wearing clothes? There is the head louse, which lives in hair. There is the pubic louse, which lives in coarse body hair. And there is the body louse, sometimes called the clothing louse, which is different from the others in a crucial way. It does not live on the body at all. It lives and lays its eggs in the seams of clothing, crawling onto the skin only to feed. That means the body louse could not have existed until humans wore clothes regularly enough to give it a home. In 2003, researchers Ralph Kittler, Manfred Kayser, and Mark Stoneking realized this made the body louse a kind of clock. By measuring how much the body louse's DNA had diverged from the head louse's, they could estimate when the two split, and therefore roughly when clothing appeared. Their first estimate landed around 107,000 years ago, and later work pushed the figure toward 170,000 years ago. If that later date holds, it means humans were wearing clothes something like 70,000 years before they migrated out of Africa into the cold of Eurasia. Since fabric and hide almost never survived the millennia, this parasite offers one of the only independent ways to date the invention. The lice have more to tell. The human pubic louse is genetically closest to the louse that lives on gorillas, implying our ancestors picked it up from gorillas, or a shared environment 3 to 4 million years ago. David Reed of the University of Florida reconstructed much of this deep coevolution, showing how the family tree of primate lice mirrors the family tree of the primates themselves, a pattern called cospeciation. Where the hosts split, the lice split too. There is even a hint that one lineage of human head lice may have been acquired from an archaic human species, perhaps Homo erectus. Lice are obligate parasites, unable to survive more than a day or two away from a human host, spread by close contact, which is why they flourish in crowded and unwashed conditions. That made the body louse one of history's great killers by proxy, because it carries epidemic typhus, trench fever, and louse-borne relapsing fever, diseases that later emptied prisons and destroyed armies. Ancient neats, the cemented eggs of lice, have been found glued to the hair of mummies from Egypt, Peru, and Roman times, and fine-toothed night combs turn up across the ancient world. Clothing was the adaptation that let humans colonize freezing latitudes, and eventually the Arctic. And we know roughly when it happened, only because a tiny insect evolved to live in the seams. Once we had clothes and had spread into every climate, we started carrying our parasites into places they should never have survived. Number 4. Hookworm and the Bering Land Bridge. A humble intestinal worm may hold a clue to one of the biggest mysteries in archaeology: how and when the first people reached the Americas. Hookworms, the species Ankylostoma duodenale and Necator americanus, are blood-feeding nematodes. Their larvae live in warm, moist soil and burrow directly through human skin, usually through the bare soles of the feet, before traveling to the intestine to feed on blood. Heavy infections cause severe anemia and stunt the growth and even the mental development of children. But the key fact is this: the free-living larvae cannot survive prolonged freezing. Cold kills them. Now, hold that against the standard story of how humans reached the New World. In that model, the first Americans walked from Siberia across the Bering Land Bridge during the last ice age, through a bitterly cold Arctic corridor that should have killed any hookworm they carried. And yet, hookworm eggs have been recovered from human coprolites, fossilized feces, in Brazil dated to roughly 7,000 years ago, with some claims reaching further back still. The Brazilian parasitologists Adaldo Araújo and Luiz Fernando Ferreira, working at the Oswaldo Cruz Foundation, documented these pre-Columbian New World finds and pointed out the paradox directly. A warm climate worm that cannot survive freezing had no business being in ancient South America if its human hosts had crossed a frozen land bridge. So how did it get there? The finding supports alternative ideas: that people entered the Americas along a milder Pacific coastal route, or arrived by sea, or came earlier and by warmer paths than the classic ice-free corridor. One debated possibility is that a fast enough crossing could let adult worms survive inside the human gut, even as the soil stage died in the cold. Either way, the parasite has become a biological argument in a debate usually fought with stone tools and radiocarbon dates. Sites like Monte Verde in Chile, at about 14,500 years old, had already pushed human presence earlier than the old Clovis first model allowed. The science that made this possible is remarkable in itself. Paleoparasitology was largely pioneered in Brazil, at the Fundação Oswaldo Cruz, beginning in the 1970s and 80s. Researchers learned to rehydrate ancient coprolites in a trisodium phosphate solution, which recovers fragile parasite eggs intact for identification under a microscope after thousands of years. There is a further twist. Necator americanus means American killer, yet the worm was long assumed to have arrived only after 1592, brought across the Atlantic in the bodies of enslaved Africans. The pre-Columbian coprolite evidence challenges that story, suggesting hookworm was already here millennia before Europeans arrived. Hookworm would go on to devastate the American South, where the Rockefeller Sanitary Commission in 1909 attacked it as the germ of laziness. If a worm can rewrite the map of human migration, imagine what a fluke could do to an entire river civilization. Number 5. Schistosomiasis in the Nile mummies. For 3,000 years, the civilization of the Nile may have been quietly drained by a worm that swims in still water. The disease is schistosomiasis, also called bilharzia, caused by parasitic flatworms, or blood flukes, that live inside the blood vessels of the abdomen and bladder. Two species haunted the Nile world: Schistosoma hematobium, which infects the urinary system, and Schistosoma mansoni, which infects the intestines. Their life cycle passes through freshwater snails. Larvae called cercariae emerge from the snails into the water and then penetrate the skin of anyone standing in it. The urinary form causes blood in the urine, and there is a haunting theory that ancient Egyptians saw this bleeding in young men as a natural rite of passage, a kind of male menstruation, never realizing it was a sign of infection. Calcified schistosome eggs have been found in Egyptian mummies more than 5,000 years old, including remains from before the age of the pharaohs. Even more striking, a 2014 study reported Schistosoma mansoni eggs in a skeleton about 6,200 years old from Tel Zaydan in Syria, and the researchers tied the infection directly to the invention of irrigation. When humans dug canals to water their crops, they created still, warm channels that were paradise for snails, and the disease spread along the water they had engineered. The written record may echo this. Ancient Egyptian medical texts, including the Ebers and Hurst papyri, describe a condition of blood in the urine, the so-called AEA disease, which many believe was schistosomiasis. Mark Armand Ruffer, working in Egypt around 1910, was the first to identify calcified schistosome eggs in mummy tissue, a discovery that helped found the modern science of paleopathology. Later researchers used the ELISA immunological test to detect the parasite's antigens in mummies, confirming infection even where eggs were scarce. The biology of the fluke is extraordinary. Males and females pair for life, the slender female lying permanently in a groove that runs the length of the male's body, an unusual parasitic monogamy. The eggs are armed with sharp spines that cut through tissue to escape the body, and this cutting and scarring causes the bleeding, and, over decades, bladder fibrosis, liver damage, and one of the leading cancers of historic Egypt, cancer of the bladder. The parasite suppresses the host's immune system and can live in the blood for decades, laying thousands of eggs. Nubian mummies from Sudan show high infection rates too, meaning the disease spanned the entire Nile corridor. And the story is not only ancient. The Aswan High Dam, completed in 1970, replaced the seasonal flood with permanent irrigation, and schistosomiasis surged. More than 200 million people are infected today, second only to malaria among parasitic diseases. But not every parasite that shaped us can be seen under a microscope in a mummy. Some rewired the mind itself. Number 6. Toxoplasma Gondii. There is a parasite that can change the behavior of the creature it infects, and it may be living in your brain right now. Toxoplasma Gondii is a single-celled protozoan that can infect nearly every warm-blooded animal on Earth, but it can only reproduce sexually in one place: the intestines of cats. Estimates suggest it infects between 30 and 50% of all living humans, one of the most widespread parasites of our species. Most never know. Its most unsettling trick appears in rodents. A rat or mouse infected with toxoplasma loses its instinctive terror of the smell of cats, and some studies describe infected rodents becoming almost attracted to cat odor, a phenomenon researchers have called fatal feline attraction. The logic is chillingly elegant. The parasite can only complete its life cycle inside a cat, so it manipulates the mouse into being caught and eaten, delivering itself to its final host. This effect was documented by Joanne Webster and explored further by Robert Sapolsky and Ajay Vyas, who showed that infected rats lose their aversion to cat urine. The manipulation seems tied to cysts forming in a fear-processing part of the brain called the amygdala, and to the parasite raising levels of the neurotransmitter dopamine. In the body, toxoplasma forms dormant tissue cysts, mostly in the brain and muscle, that can persist for the rest of the host's life. Humans usually catch it by eating undercooked meat that contains these cysts, or by swallowing hardy egg-like oocysts from cat feces lurking in soil, water, or unwashed produce. For most healthy people, it causes little more than a mild flu, but it is dangerous during pregnancy, where it can harm the developing fetus, and in people with weakened immune systems, where it can cause lesions in the brain. More controversially, some studies have found statistical links between chronic infection and slower reaction times, greater risk-taking, more traffic accidents, and even schizophrenia, though whether the parasite truly causes these effects is fiercely debated. What makes Toxoplasma a true deep-time ruler is its ancient lineage. Its genus belongs to a group of parasites called the Apicomplexa, which also includes the malaria parasite, and these lineages diverged hundreds of millions of years ago. Because cats are the definitive host, the parasite's history is bound to the evolution of the cat family, stretching back tens of millions of years. Our own ancestors, living alongside wild cats across the whole span of human evolution, would have been exposed the entire time. It was first identified in 1908 in a small North African rodent called the Gondii by Charles Nicolle and Louis Monceau. The parasite crosses the blood-brain barrier by hijacking the host's own immune cells like a Trojan horse, and a single infected cat can shed hundreds of millions of oocysts that survive in soil for over a year. It even kills sea otters when oocysts wash from the land into the sea. Whatever it does to human minds, it has been an invisible companion of humanity since long before the first farmer planted the first seed, and it was that first farmer who unleashed the real explosion. Number 7. Whipworm and the Neolithic explosion. When humans invented farming, they also invented a golden age for their parasites, and no worm tells that story better than the whipworm. Trichuris trichura takes its name from its shape, a long, thin, thread-like front end and a shorter, thicker rear that looks like the handle of a whip. The adults burrow into the wall of the large intestine, and a single female can release between 3,000 and 20,000 eggs every day. Those eggs have a distinctive lemon or barrel shape with clear plugs at each end, which makes them easy to recognize in ancient samples even after thousands of years. The whipworm's egg needs warm, moist, shaded soil to develop, and it spreads by the fecal-oral route, which is a polite way of saying it thrives wherever people live, crowded together near their own waste. For most of human history, mobile foragers avoided this trap because they moved on and left their contamination behind. But the Neolithic transition to farming and permanent villages changed everything. People settled down, built houses, stored grain, penned animals, and stayed put beside their own refuse. The result was a documented surge in whipworm and other soil-transmitted worms. Whipworm eggs are among the most common parasites recovered from ancient latrines, cesspits, and coprolites across Europe and the Near East. Even Ötzi, the Copper Age Iceman who died in the Alps around 5,300 years ago, carried whipworm. The archaeologist Piers Mitchell of the University of Cambridge has led major surveys showing that parasite diversity rose alongside the spread of farming, towns, and even Roman sanitation. That last part is the great surprise. You might expect the Romans, with their famous baths and public toilets, to have fewer intestinal parasites than the Iron Age peoples before them. Instead, they had as many or more for grimly practical reasons. Romans fertilized their crops with human waste, spreading eggs across the fields, and ate a fermented fish sauce called garum that could carry parasites across the empire. At settlements like Çatalhöyük, parasites are tied to living closely with penned animals and with rodents drawn to stored grain. Scientists call this whole shift the first epidemiological transition, the great surge of infectious and parasitic disease that came with agriculture, the hidden price of civilization. Heavy whipworm infection rarely kills outright, but it causes chronic diarrhea, anemia, stunted growth, and in children, even rectal prolapse. Researchers recover the eggs from the pelvic soil of ancient burials, where the contents of the gut settle after the body decays, and they have even extracted ancient DNA from the eggs themselves to trace the parasite's own history. The eggs can stay viable in soil for years, which is why breaking the cycle demands sustained sanitation. Whipworm still infects hundreds of millions of people today, overwhelmingly children. But of all the parasites that came for the farmers, one had already been shaping our very blood for far longer. Number 8. Malaria and the human genome. No parasite has killed more human beings than the one that causes malaria, and none has left a deeper mark on what we are. Malaria is caused by single-celled Plasmodium parasites, carried by female Anopheles mosquitoes, and the deadliest species that infects humans is Plasmodium falciparum. Many researchers believe malaria is the single deadliest disease in all of human history, plausibly responsible for a significant fraction of every human who has ever died, and its pressure has been so relentless over so many thousands of years that it has physically rewritten the human genome. The most famous example is the sickle-cell trait, a single mutation in the gene for hemoglobin, the oxygen-carrying molecule in red blood cells, protects the carrier against severe falciparum malaria. The catch is that inheriting 2 copies of the mutation causes sickle cell disease, and yet the gene remains common in malarial regions, because carrying 1 copy is a survival advantage. This is the textbook case of what biologists call balancing selection, where a harmful gene is kept common because it saves lives in a particular environment. Malaria drove other adaptations too. The thalassemias, a deficiency in an enzyme called G6PD, and the hemoglobin C and E variants. In much of West and Central Africa, nearly everyone carries the Duffy-negative blood group, which protects against Plasmodium vivax by removing the doorway the parasite uses to enter red blood cells. Anthony Allison proved the link between sickle cell and malaria in East Africa in the 1950s, one of the founding discoveries of evolutionary medicine. Where did such a deadly parasite come from? Plasmodium falciparum appears to have jumped to humans from gorillas, and its closest known relative still infects wild gorillas in Central Africa. Molecular estimates suggest the parasite's great expansion in humans intensified within roughly the last 10,000 years, and the timing is no accident. When people cleared forests to plant crops, they created sunlit pools of standing water, the perfect nursery for the mosquito Anopheles gambii. Farming did not just unleash the whipworm, it supercharged malaria. The parasite's life cycle is complex, moving through liver and blood stages inside the human body, and reproducing sexually in the mosquito's gut. And Plasmodium vivax can lie dormant in the liver as hidden forms called hypnozoites, causing relapses months or years later. Ancient DNA has recovered Plasmodium from Roman era and medieval skeletons, and the remains of the pharaoh Tutankhamun tested positive for falciparum DNA, showing the disease reached even royalty. The Greek physician Hippocrates described its intermittent fevers and linked them to marshy ground, and the name malaria comes from the medieval Italian mala aria, meaning bad air, from the old belief that swamp vapors caused it. Malaria still causes more than 200 million cases and roughly 600,000 deaths every year, mostly African children under the age of 5. Billions of people alive today carry DNA that was shaped by this parasite before a single word of history was written. And where malaria wrote itself into our blood, other worms wrote themselves into the very dirt of our settlements. Number 9. Ascaris Roundworm. If you want to read the sanitation history of a lost people, you dig beneath their toilets and count the eggs of Ascaris lumbricoides. This is the giant human roundworm, and giant is the right word. Adult females can reach 20 to 35 centimeters long, making it the largest common intestinal worm of humans. A single female produces around 200,000 eggs a day, and those eggs are extraordinarily tough, wrapped in a thick, warty outer coat that lets them survive in soil for years and preserve beautifully in archaeological sediment. That durability is why Ascaris is one of the most frequently recovered parasites from the ancient world. The worm spreads by the fecal-oral route. A person swallows the eggs from contaminated food, water, or soil. The eggs hatch in the gut, and the larvae take a strange detour, migrating through the lungs before being coughed up and swallowed again to mature in the intestine. Ascaris eggs have been found on every inhabited continent, from Peruvian and Chilean mummies to the medieval cesspits of European cities. Some of the oldest finds include Ascaris-type eggs in coprolites from the Americas thousands of years old, and eggs in Bronze Age and Iron Age latrines across Europe. Because the worm depends on crowding and poor waste disposal, The abundance of its eggs in a site is a direct index of how densely people lived and how they handled their sewage. There is a domestic twist to its history. The pig roundworm, Ascaris suum, is nearly identical to the human species and can cross between the two, tangling the parasite's story with the domestication of pigs. Heavy infection in children causes intestinal blockage, malnutrition, and stunted growth, and a writhing mass of worms can physically obstruct the bowel, while larval migration through the lungs can trigger a pneumonia-like illness called Loeffler syndrome. The worm was formally described by Linnaeus in 1758, but was known to physicians in antiquity, appearing in Egyptian, Greek, and Chinese medical texts. Modern science reads these eggs like a ledger. Researchers count eggs per gram of sediment to estimate how intense infection was in a past population, and they have even sequenced ancient DNA from the eggs to trace the worm's spread along the routes of migration and trade. Roman period latrines across the empire consistently contain Ascaris, and medieval cesspits in cities like York, Lubeck, and Amsterdam are rich in both Ascaris and Whipworm eggs, documenting the grim sanitation of early urban life. When both worms appear together, it is a reliable sign that human waste was contaminating the food supply. Ascaris is one of the soil-transmitted worms the World Health Organization still targets with mass deworming, and it infects between 800 million and 1 billion people, a living thread connecting an ancient latrine to a modern clinic. But one worm followed humanity even more faithfully than this one, so faithfully that it can trace our footsteps across the planet. Number 10. Pinworm, the migration tracker. The last parasite in our countdown is small, nearly harmless, and one of the most useful tools science has for reconstructing where prehistoric humans traveled. Enterobius vermicularis, the pinworm or threadworm, is a slender white nematode whose females grow only about 8 to 13 millimeters long. What makes it special is its loyalty. Pinworm is one of the most human-specific parasites known, infecting essentially only humans and traveling only where humans travel. Where you find ancient pinworm, you have found ancient people. Its life cycle is unusually direct. The female pinworm migrates out of the anus at night to lay her eggs on the surrounding skin, which causes intense itching and makes the parasite spread easily from person to person, especially among children. Because it is so tightly bound to us, its eggs in ancient feces become powerful evidence of human occupation and of the paths people took. Pinworm eggs have been recovered from coprolites in the Americas dated to well over 10,000 years ago, among the oldest evidence of any human parasite in the New World. Some of the oldest American finds come from sites in the western United States, including Danger Cave and Hog Up Cave in Utah, as well as coprolites from South America. Here is where pinworm becomes a detective. Recall the hookworm, which could not survive a frozen crossing of the Bering Land Bridge. Pinworm faces no such problem because it lives entirely inside the human host and never needs a stage in cold soil. So it could ride across a frozen Beringia with no trouble at all. The contrast between the two worms is exactly what makes them useful. Pinworm's presence and hookworm's absence in the earliest sites help constrain the possible routes and timing of the first Americans. The paleoparasitologists Adauto Araujo and Carl Reinhard used exactly this kind of reasoning to reconstruct how the Americas were peopled. Carl Reinhard of the University of Nebraska is considered a founder of archaeoparasitology in North America and pioneered many of the techniques for analyzing coprolites. The parasite's egg is distinctive, asymmetrical, and flattened on one side, recognizable under the microscope even after thousands of years. Because pinworm needs no intermediate host and no soil development stage, it functions almost like a genetic tag that humanity carried out of Africa and pressed into every continent it reached, its lineages tracing how ancient populations split apart and moved. In daily life, it is a minor nuisance, causing little more than nighttime itching, diagnosed today with a piece of tape pressed to the skin to capture the eggs laid overnight. Yet it remains the most common worm infection in the temperate developed world, including the United States and Europe. Together, the coprolite worms, the pinworm and the hookworm, the whipworm and the roundworm, form a biological archive of where humanity went and what it carried the whole way. And when you step back from these 10 stories, a single pattern begins to emerge. Look across these 10 parasites, and 2 moments keep surfacing as the great turning points in human disease: settling down and farming. For hundreds of thousands of years, humans lived as mobile foragers, and mobility was a kind of medicine. When a camp grew filthy, people simply moved, leaving the contaminated ground and their parasites behind. The whipworm, the roundworm, and the pinworm all depend on the fecal-oral cycle, and a wandering band breaks that cycle every time it packs up and leaves. Then, roughly 12,000 years ago, that escape hatch closed. People built permanent villages and stayed. They lived beside their own waste, stored grain that drew rodents, and penned animals against their houses. The contamination that foragers outran now surrounded settled farmers day and night, and reinfection became constant. Whipworm and Ascaris eggs pile up in the latrines and burials of exactly these communities. Scientists call the result the first epidemiological transition, and it is one of the great ironies of human history. The very inventions that let our population explode, agriculture and permanent settlement, also detonated our burden of disease. Farming struck a second, subtler blow through the landscape itself. When humans dug irrigation canals along the Nile and across the Near East, they made still warm water that was perfect for the snails carrying Schistosomiasis, and the blood flukes spread along the channels people had engineered to feed themselves. When they cleared forests for fields, they opened sunlit pools where malarial mosquitoes bred, and malaria intensified in the same window. The same act, again and again, brought more food and more parasites in the same breath. Chagas disease is the important counterexample. It afflicted the mobile Chinchorro foragers of the Atacama long before any city rose, because its insect vector followed people into even their simplest shelters. Parasites did not wait for civilization, but civilization threw open the doors, and a whole invisible population came flooding in. The astonishing part is that we can reconstruct any of this, and the tools that let us do it are stranger than the story itself. Consider what it takes to prove that a worm crossed an ice age. The parasites in this video are not just villains. They have become witnesses, and learning to read their testimony created an entire science. Paleoparasitology, the study of ancient parasites, was pioneered in Brazil at the Fundação Oswaldo Cruz in the 1970s and 80s, and one of its foundational techniques is almost humble. Researchers take a coprolite, a piece of fossilized feces, and rehydrate it in a solution of trisodium phosphate. After thousands of years dry, the ancient tissue softens, and the durable eggs of parasites float free, ready to be identified under a microscope by their unmistakable shapes. The plugged lemon of the whipworm, the warty shell of the roundworm, the flattened asymmetry of the pinworm. From those shapes come astonishing deductions. The logic of pinworm versus hookworm is the clearest example. Pinworm lives entirely inside the human body, so it could survive a frozen crossing of Beringia, while hookworm needs warm soil and could not. So when hookworm turns up in ancient South America, it whispers that not everyone came the frozen way. This is how parasites became an independent check on archaeology, a biological argument running alongside stone tools and radiocarbon dates. Adalto Araujo and Carl Reinhard built careers on this reasoning, and Reinhard, at the University of Nebraska, helped turn coprolite analysis into a rigorous method. The lice tell a different kind of story, acting not as a map, but as a clock. Because the body louse can only live in clothing, the moment it's split from the head louse marks. roughly the moment humans began wearing clothes, an event that leaves almost no other trace because fabric rots away. Measuring that genetic split, let researchers date an invisible invention to somewhere between 80,000 and 170,000 years ago. This is the quiet power of parasites as historians. They preserve the unfossilizable. They recorded when we dressed ourselves, when we crossed into new worlds, when we dug our first canals, not in stone or ink, but in their own bodies and their own DNA. And some of them recorded that history in a place even more intimate, inside us. The deepest mark these parasites left is not in the soil or a museum drawer. It is in the human genome, carried by billions of people who will never know the war their ancestors fought. Malaria is the clearest author of these scars. Because it killed so many children before they could reproduce, any mutation offering even partial protection spread rapidly, no matter its cost. The sickle-cell mutation is the famous result, common across malarial regions, because a single copy shields against the parasite, even though a double copy causes disease. This is balancing selection in action, a harmful gene held in place by an even deadlier threat. Malaria's signature is written in many places at once. The thalassemias, the enzyme deficiency called G6PD, the hemoglobin C and E variants, and the Duffy negative blood group that guards against Plasmodium vivax are all adaptations shaped by the same relentless pressure. Anthony Allison first connected sickle cell to malaria in the 1950s, and in doing so helped found the field now called evolutionary medicine, the study of how disease sculpted our biology. To read your own blood is, in part, to read a record of the parasites your ancestors survived. Other parasites left subtler fingerprints. Toxoplasma Gondii demonstrably alters rodent behavior by forming cysts in the brain and shifting dopamine levels, and researchers still argue over whether its lifelong presence nudges human behavior as well. The lice, though they did not rewrite our DNA, left a genealogical record so faithful that their family tree mirrors ours, preserving the memory of an ancient host switch from gorillas. The lesson is humbling. The human body is not a finished design handed down intact. It is a battlefield, shaped move by organisms we could not see, in a war that began before language and has never truly ended. And for all we have learned, the most honest thing to admit is how much remains unknown. For every confident date in this video, there's a live argument behind it, and the honest frontier of this science is crowded with open questions. Take the origin of clothing. The louse clock gives an answer, but that answer swings from around 80,000 to 170,000 years ago, depending on the assumed mutation rate. Molecular clocks are powerful, but they are only as reliable as the tick rate you feed them, and that rate is genuinely uncertain. So the true birthday of the first garment remains blurry by tens of thousands of years. The hookworm paradox is not fully settled either. Its presence in ancient South America strongly suggests not everyone crossed a frozen Beringia, but researchers still debate whether a fast enough crossing let adult worms survive inside human hosts, even as the soil stage died. The peopling of the Americas is a moving target, and the worms are one line of evidence among many that do not yet perfectly agree. Toxoplasma raises perhaps the most provocative uncertainty of all. It clearly manipulates rodents, and the statistics linking it to human behavior are intriguing, but whether it meaningfully shapes human personality across whole populations or is simply correlated with other factors remains genuinely unresolved. There are deeper unknowns still. We do not know how far back the human histories of Chagas and Toxoplasma truly run, only that their wild reservoirs are far older than our species, and the archaeological record itself is biased. The durable warty eggs of Ascaris and the plugged eggs of Whipworm dominate the samples, while soft-bodied parasites and single-celled protozoans leave far fainter traces. So our picture is skewed toward the worms that fossilize well, Ancient DNA offers a way forward, but carries its own peril: the constant risk of contamination by modern organisms. None of this is a weakness of the science, so much as a sign of how young and how alive it is. Every rehydrated coprolite and every sequenced egg redraws the map a little. Which brings us back to the largest idea of all, the one that has been lurking behind every item on this list. Before there were kings, there were parasites. Before writing, before cities, before the first name was ever carved into stone, an invisible empire already governed the human world. It decided where people could live, because a valley thick with malarial mosquitoes or schistosome snails could break a settlement before it began. It decided how long they lived, draining the strength of Nile farmers and hollowing the hearts of Atacama fishermen. It decided, in the most literal sense, what they became, editing the human genome, one deadly generation at a time, until protection against malaria was written into the blood of entire continents. These organisms ruled without armies or monuments. The Guinea worm may have coiled itself into the oldest symbol of medicine. The clothing louse recorded the day we first dressed ourselves. The pinworm and the hookworm traced our footsteps across the land bridges and coastlines of a new world, keeping a diary in fossilized feces we are only now learning to read. And the malaria parasite fought a war with our species so long and so brutal that its scars are carried today by billions who have never seen a mosquito net, let alone a swamp. The strangest part is that this empire never fell. Toxoplasma still lives in perhaps a third of all humans. Whipworm and roundworm still infect hundreds of millions, and malaria still kills a child roughly every minute. The Guinea worm hangs on by a thread of fewer than a dozen cases a year, and if it finally vanishes, it will be the first human parasite we ever deliberately erased from the earth, a single victory against an empire that has ruled us since before we could remember. We tend to tell the human story as a tale of what people built and conquered, but the oldest chapters of that story were not written by human hands at all. They were written by the things that lived inside us, in our blood, our guts, and our genes, long before anyone thought to write anything down.